Wow, this is going to be hard act to follow, very hard.
And here,
everybody bring expectations down.
I would argue with that. Actually, I think we're very lucky to have one of Israel's greatest entrepreneurs here with us today. And we're now going to move from the future of food, although that's something that we will learn in a minute that the professor can very well address, to another topic that impacts human health, and that is the future of drug development. So I'd like to first, kind of set the stage by introducing Professor Yaakov Nahmias. He is the director of the Center of Bioengineering at Hebrew University. He is the founder and president of Future Meat, an Israeli food tech company that went from 0 to $1 billion valuation in 4 years. Founder and CSO of Tissue Dynamics, a spin-off of Hebrew University, and a fellow of the Royal Society of Medicine in Britain. So, as a journalist, an editor-in-chief of The Innovator, I write about AI's impact on drug development and medicine in general, and we've all heard about how AI has helped us sequence the human genome. And it also now today helps radiologists read X-rays. And this is all great and has helped advance medicine, but there's still a big gap. And as the professor explained to me, that gap is between genomics and pathology. In the middle, we have human biology, which is chaotic, unreliable, and very difficult for AI to read. So we have not advanced as far as we might in actually applying AI to drug development. So let's start by talking about the challenges. Where are we today? How long does it take to develop drugs? How much does it cost? And where do we go from here?
So thanks for the introduction, for both of you. Drug development is a really Sisyphean process, and it costs about $2.6 billion to bring one drug to the market, and takes, on average, about 12 years. And then if people imagine some professor in the laboratory, sitting for 20 years on a single molecule and then taking it to a mouse model and then bringing it to the clinic, that's not how it works today. We start with libraries of 500,000, 2 million compounds, and then we screened all of them against Petri dishes, right? Mm-hmm. And then we don't find one molecule for breast cancer, we find 5,000. And then the process is taking those molecules, going to animal models, and trying to find, out of those 5,000, which molecule is the most effective and the most safe at the same time, so the least toxic. And this is the molecule that you take to a clinical study. The problem is that after you did all of these experiments on hundreds of animals, you go to a clinical study and 88% of the cases simply fail. It's worse than throwing dice. You can get better odds in a lot of other places. And even those drugs that manage to get FDA approval, another 10% of them actually fail outside on the market. It's a huge issue, and the most important thing is that it's because of our reliance on small animal models. So mice and rats are not human. They have different genetics, they have different metabolism, and they have different physiology. So a lot of the things we study in animals simply do not translate. It's easy to treat cancer in rodents. It's easy to treat diabetes in rodents. None of these therapies translated. That's probably the most important hurdle. The second problem is that once you fail in a clinical study, you usually don't understand why you failed. Mm-hmm. You have to go back to the beginning and take another molecule and try again, and fail. And then go back to the beginning and take another molecule and try again. So if you think about Sisyphus actually pushing the rock up the mountain again and again and again, that's drug development today.
Okay. How are advances in technology helping to change that? Can we start with maybe living tissue or chip, organs on a chip?
Right, so about a decade ago, we were one of the first laboratories to actually introduce organ on a chip. So the first one was liver on a chip that was created by Mike Schuller, and I helped popularize it in URL. But the concept is to take human cells that have human genetics, create out of them these three-dimensional tissues, and we can make right now liver, heart, brain, kidneys, very, very tiny, about 0.5 millimeter in size, all human tissues. So they have human genetics, human metabolism, and then you put them in a microfluidic environment that actually mimics normal human physiology. Mm-hmm. And I know it sounds like science fiction, but this is a decade-old technology. And it allows us to essentially get much more predictive than animals.
Okay. But how do you read the data from this?
Yeah, so this is where things really took off for us, because you used to treat those chips exactly like you treat animals. So in an animal experiment, you give an animal a drug, you wait a few days, then you kill the animal, you open it, and you see what happened. And people used to treat those microfluidic chips in the same way. They still do in many places. So you give the drug, you use the chip, and then you look at it, and you get a yes-no answer. Mm-hmm. Yes and no doesn't help me. I need to understand why and how. So what we did about 5 years ago is start integrating sensors, optoelectronic sensors, inside human living tissue. And essentially what we made are bionic microorgans, as we like to call them. And we can communicate with them very, very rapidly. So it's just like having sensors in your car, right? Okay. So if you're driving your car, you have a sensor for air pressure in your tires, for oil, for temperature of your radiator. Before your car burns up in flames on the Autobahn here in Germany, you know that something is wrong, mm-hmm. because one of those sensors would tell you what is wrong, and you can actually decide how to fix it. Okay. And this is exactly what we did with those tissues more than 5 years ago, and enabled us to really break through into mechanism of action, making this Sisyphean process an iterative one. So you take a drug, if you see that it doesn't work, you understand exactly why, and you can fix it. So your next step is already going to succeed. One of the biggest stories was a drug that was developed by Pfizer called troglitazone, which was it was safe in animals, it was safe in clinical studies, and when we got it out to the market, essentially it caused liver damage in 1 out of 30,000 patients and had to be withdrawn, costing billions of dollars to the company. And our platform could very, very rapidly detect it. We could actually see what was wrong in a way that no other technology was able to do that. And that was more than 5 years ago.
And so by just tweaking something, you would have been able to prevent these deaths?
That's right.
Okay. So in your car, if we continue that analogy, of having the sensors telling you something wrong, in cars, the next step is autonomous. So can we make, all of this, do the equivalent? Can we make this automized by AI?
Exactly. So this is where it becomes really exciting. So about a decade ago, I moved back from Harvard to the Hebrew University, and my office was right next to Amnon Shashua, the founder of Mobileye. And exactly in the same way that sensors in our car enabled us to think about autonomous driving, right? How do we make the car work on its own? We can start thinking about drug development process being completely autonomous. So the company that I founded, Tissue Dynamics, does exactly that. Together with Beckman Coulter, we built this large robotic platform that can handle 20,000 human microtissues with sensors all at the same time. And then the robotic system actually makes the organs, it can test the molecules, it can identify the one that works, and it does safety assessment exactly at the same time, and the entire lead optimization and the chemistry essentially taking it step by step all the way to product. It's going to be a fun couple of years when we finish building the platform, but this is really where we want to go. This death valley that is between genomic and computational biology, where AI is doing amazing work, and pathology, where image processing and AI is doing amazing work. This exactly is the death valley in between, where biology is messy, like you said before. There's variance between cells, there's variance between technicians, there's differences between microscopes, between antibodies, between images. Couple of major pharmaceutical companies that were trying to use AI exactly in this area a few years ago weren't able to, even though they spent tens of millions of dollars. We think this robotic platform that we're developing would enable us to do exactly that.
So let's talk about where your company is in its trajectory. So you were working with some pharma companies now in trials. Can you tell us a little bit about that?
Yeah, so early on in COVID, we actually identified a metabolic pathway that was actually shut down by a virus replication. We did it with Mount Sinai Hospital, and then we very quickly saw that Abbott had a drug on the market that could act with the same pathway. We demonstrated that. It took a ton of, we're in Israel, so we have a lot of access to HMO data. Israeli healthcare is digitized.
Right.
So we could very, very rapidly see that the pathways are there, validate it with patient data, and then go to a phase 3 clinical study. The open label data was great, but now we are just finished recruiting our phase 3 last patient. So the proof needs to be in the pudding. We need to see the placebo-controlled data. But one of the most interesting things I think that we did last year was go through an entire process of drug development that was animal-free. We developed a model for the kidney and specifically look at chemotherapy-induced injury in the kidney. And the signal that we saw was a metabolic fingerprint that if you wake me up and say in the middle of the night like my kids do and ask, Daddy, Daddy, what is this? I would tell you, this looks like fatty liver disease. Mm-hmm. It has the same hallmark of fatty liver disease, but it wasn't. It was actually a metabolic signature in the kidneys. And because we could make that connection that an AI could, we could very rapidly identify the pathway and come up with a solution, essentially a combination therapy that, again, we use clinical data to validate very, very rapidly. This entire process was 12 months, and it was the cover of Science Translational Medicine.
That sounds pretty impressive. So your company is still pretty young, though, right? So you're about to go out for your Series A, looking for some investors so you can scale up. If you manage to prove this and you scale, what will be the impact of this? First, in terms of economics, and speeding up drug discovery, and then secondly, on healthcare in general.
So this dramatic loss of success in clinical studies, 88% and another 10% out in the market, is really the main problem. We think we can reduce the entire process, the risk in the entire process by about 85%. Wow. That would reduce the cost of drug development from $2.6 billion to a drug to somewhere on the order of $500 million per drug. This could be transformative because today, even though there are thousands of small pharmaceutical companies, none of them would reach the market. All of them have to be sold to a few mega corporations that can actually have the funding to support what you need for phase 3 studies and reach the market. And if you manage to dramatically reduce the cost and reduce the risk, you will have not 5 or 10 mega corporations. You'll have 5,000, 50,000 companies that can do that. And that could completely change the ecosystem. You can start thinking about orphan diseases. You can start thinking about massive repurposing efforts for diseases in sub-Saharan Africa, or specific personalized medicine to specific sections in South America, India, China, where speed is of the essence.
So are we really talking about democratizing healthcare?
That's going to take time.
Okay, what do you think the timeline might be?
You know, they say that prophecy is given to fools, and in spite of what my wife says, I'm less one. It's very hard to predict how fast AI would move, and what we really need to do is to put the platforms together that would enable it to be successful. I think the lack of reliable, repetitive data in biology has hurt its application up till now. But robotic systems like the one that Tissue Dynamics is building and other companies that are doing amazing work in that area could really fundamentally transform the field by allowing many people to apply artificial intelligence and machine learning tools to fundamentally change the way we do biology.
That's no small feat, and you heard it here first. You know, it sounds like, you know, if this works, it could really revolutionize drug development. So let's give a huge round of applause to Professor Nahmias.
Thank you. And if everybody make sure to wash your hands, I don't know how many people heard about the monkeypox that is running around. We should really look at everything very, very carefully as as things progress.
Okay, thank you very much.